So one really fascinating class of peptides that I've been interested in are fragments of full-length proteins. These protein fragments have some inherent capability to actually bind to the native targets of their full-length proteins. And if they do that, they can potentially bind to the correct target, but lack other features of the full-length protein, and in this way act as an inhibitor. In the classic biology sense, this might be called a dominant negative inhibitor. And so I was really interested in the ability of small peptide fragments of proteins to perform this kind of function. Historically, this idea has been developed largely through naturally occurring truncation mutations and splice variants that can lead to proteins having, or variants of proteins having a dominant negative effect. But I wanted to see how generalizable this was and to what extent just small peptide fragments of a protein could actually do this and provide us with a tool to both study and control biology. And this would really be enabled by an approach in which we could actually make high throughput measurements of many or even all of the fragments of a protein and their ability to inhibit its interactions in parallel. So we developed two complementary ways to go after this problem. One is high throughput in vivo experimental measurements in living cells, where we actually put libraries of these protein fragments directly in cells and read out their functional consequences. And the other is massively parallel computational predictions using AI methods to actually predict the ability of these fragments to inhibit interactions by predicting their binding interactions.